what I want to do in the next few minutes is tell you about how the work that's been done in Sunderland over the last many years could be a solution to the problem of taking cysteamine. I don't really need to tell this audience what the problems are when taking cysteamine, but my rare diseases clinic developed, and I've looked after about 20, 30 patients with cystinosis. I've yet to meet an adult cystinosis patient that takes their cysteamine 4 times a day. Any patient that turned up in my clinic and sat there and said, I take my cysteamine 4 times a day, I'd send them out and ask them to come back in when they told the truth. And you all know about the bad breath, the halitosis, and you all know, I'm sure, about the gastritis. And the fact that in the UK, you need to repeat your prescription every 7 days. And so we've developed a prodrug. Now, what is a prodrug? They've been around for decades. A prodrug is a drug which is inactive. When you take it in the tablet, it gets into your body, and a number of chemical reactions will take place in your body that make it active. Many antibiotics actually are prodrugs. And there are quite a lot of other drugs that affect your heart and your breathing, which are prodrugs. And this way of producing drugs increases the ability of the body to absorb the drug, and it can improve the way that the body handles the drug. And it's a very, very well established principle. So I don't want to bore you too much with science, but the way that the late Ros Anderson developed this was, the red you can see in the middle is cysteamine. It's a stuff that you take either as Procysp or as Cystagon, or if there's any other makes over here, I don't know. And what was done is this. Thioester, which is a small molecule, is attached to the cysteamine, and it sits on the cysteamine where the sulfur molecule is. And it's a sulfur molecule that is released and metabolized in the body that causes the production of volatile phenols and others, which causes the bad breath and the body odor. So by putting a thioester on there, you stop it. And it also stops you getting the gastric irritation. The blue bit is is it blue? Is a sort of blue right at the other end, the ester. That improves the way the drug gets into the body, gets in very, very quickly. And the glutamate, that is the real thing. Now, I don't want to go too much into this, but in every human cell in the body, virtually every human cell in the body, including in the eye, there is an enzyme called gamma-glutamyl transpeptidase. It's actually mostly found in the liver, but every cell has it, and it's very, very important. And what you do is you put it onto the cysteamine, and when this prodrug, CF-10 we've called it, gets into the blood, the thioester and the ester are removed. And that gamma-glutamate there, it sticks to the cell. And the cysteamine gets into the cell, so it never floats around in the blood, so it never can be metabolized, release the sulfur, and cause all the side effects. That's the theory. So does it work? Right, so this slide is very simple. Basically, the little dots on the top are CF-10, and the squares on the bottom line are ordinary cysteamine. And you'll see here that both drugs get into the blood very quickly, but you'll see also that the CF-10 is about 10 times lower in the concentration than the same amount of cysteamine. So basically, when you take the CF-10, and this is all done in animals, of course, and also in cells grown in culture, you will see that for equal amounts, you will get much less in the blood of CF-10 than you would do of the ordinary cysteamine. However, it's not the blood that's important. Where does cysteamine work? It works in your kidney, in your thyroid gland, in your pancreas, and all over the body, in your eye, in your skin. And this is some work done that shows that if you look at the top line, that is the amount of cysteamine in the kidney when you have taken CF-10. And you'll see that it's much higher, much more in the kidney compared to the plasma. So there is much more CF-10, much more cysteamine in the kidney than you would do if you just took ordinary cysteamine. All this work that you've just seen proved to those of us working it, certainly to prove to Ros Anderson that this was something to take forward. Now, speaking to this audience, it's important to know that all the work that went into this, and it's called CF-10 because it was the 10th molecule of a prodrug that Ros actually worked with. So there were 9, there was CF1 to 9 that went and they didn't work. And all this work, which took place over more than 10 years, was funded entirely by Cystinosis UK. So it was the patients, your UK equivalents, who funded all this, and I think I'm going to refer to that again shortly. However, the mouse model of cystinosis is rubbish. It doesn't really look like what a patient would have. Not that you were mice, of course. Jennifer Hollywood, I think will be referred to again, is from Ireland. She then went over to New Zealand for a while. She's back in Ireland. She's currently on maternity leave, but she used modern genetic methods to produce a rat model, and you heard in the last talk about CRISPR. So this is a way of basically surgically or molecularly surgically engineering DNA, and what she did was with her team, she produced rats where the copy of the cystinosis gene was basically taken out. And so she produced a model of cystinosis when, from a pathology point of view, was exactly the same than the pathology that patients had. And so at this stage, before Jennifer got involved, we had this information from the animal work and the cell work. And basically, CF-10 and cysteamine were exactly the same. So it was just as effective. But the difference was that the evidence that we were beginning to see was that the side effects were not there. So when you, mice don't vomit, by the way, but when you gave CF-10 to mice and you looked at their stomachs, they didn't have ulceration with huge doses. And we subsequently learned that you can give 4 or 5 times the amount of CF-10 compared to cysteamine and not have side effects. And I'll come on to that in a minute. So Jennifer and her team did some work to confirm this, and then we had to produce results to show the regulators, those people that decide whether we can do clinical trials, that we had a product that was safe and effective, etc. And we then went on to do various studies. and I'm summarizing them. So we got a grant of one from the Medical Research Council, which is sort of like your NIH, but better and better run. And it has a budget of many billions of pounds, and you have to apply for grants. It's government money. And we got a grant of just over £1 million to do the regulatory work, which was done in the rat model and in the dog. And this is the summary that we got. First of all, we observed no side effects in terms of the heart and the lungs and all that sort of stuff. It didn't cause cancer gene mutations, but we found a couple of things. One, it did not cause gastritis until you gave something like 10 times the dose of equivalent cysteamine, so huge doses, the sort of thing you wouldn't give to a patient unless you wanted to kill them off. The other thing we did, we collected the breath of the dogs, and there's a new machine that we managed to obtain where you could take the breath of the dog, put it in the machine, and it would measure the amount of the volatile gases that cause the halitosis, the bad breath. Now, those of you who keep dogs will know that dogs' breath smell anyway, so it compensates for that. It doesn't actually, it does measure what the dogs put out normally, but it can separate the nasty stuff, the methanol and all those sort of things. And the other thing we found in these preliminary studies was that compared to cysteamine, normal cysteamine, the half-life, in other words, the length of time that the drug stayed in the body was much longer. And that would mean that if you translate, if the dog and the rat are similar to humans, that means that you wouldn't have to take the CF-10 more than twice a day, but probably also only once a day. That still needs to be seen. So that's what we did. And this just summarizes that. So we have a drug that gets into the tissues much better than cysteamine, about 10 times better. We have something which is well tolerated, doesn't cause side effects, it doesn't seem to cause gastritis, it doesn't cause bad breath, and in the black at the bottom, you'll see we can actually make the stuff. So you have to prove to the regulators that you can make enough to do many clinical trials, because there's no point in doing a clinical trial if you can't give the stuff to the patients. Where are we? So we're with the middle blue line. So we are just waiting to hear. In fact, it's holiday time and I'm here, and most of the team involved with this are actually away. So sometime shortly we will hear whether we've been successful with our application again to the Medical Research Council. So it's not being funded by pharmaceutical companies for a £5-point-something million grant, so we can do what's called a phase 1 study. This is the first time it'll be given to patients. Little change. So if you look at the bottom of that slide where it says CF-10 clinical trials, we've put in the money for the phase 1 study, which I'll show you in a minute. But there's a few things which we're going to do in this clinical trial which haven't been done before. One is we're actually going to measure human breath, volatile liquids. It's only been done once before in humans, and that's Herbie Newell, who's the leader of our team, Professor Newell. He put it on himself just to find out how it worked. But it hasn't been used properly in a clinical trial, so we also we don't want to rely on the amount of cysteine in your white cells as a measure of efficacy, because we think that because of the way the drug works, there'll be a lot more CF-10 in the tissues than there will be in your white cells. And so we're going to do skin biopsies take a little, a punch, a point, half percent, so 0.2, I'm trying to, I can't put it into inches, about a 0.2 or 3 of a centimetre. So what's that? I don't know. A very small amount anyway. It's about twice the size of the pin of the head of a match. Take those out, mush them up, and measure cysteine. And in order to make sure all this works, and one or two other things which I won't bore you with, we're going to do a pilot study very shortly, probably starting in September, October, where we're going to take patients, just 4 patients, 3 or 4 patients with cystinosis, and we're going to run the whole clinical trial, but using cysteamine rather than CF-10, so that we will make sure there are no logistical problems, and that we can actually measure the amount of volatile liquid, volatile gases, etc., and that skin biopsy measurements work. I'm nearly finished. And so what we're going to do is we're going to do a phase 1A and a phase 1B. So the first bit is we're going to give it to a few patients, probably about 6, in increasing doses, because we think we know the dose, but we have to show the regulators that what the maximum dose is, what the minimum dose is, and we reckon that the dose will be at, once or twice a day, as I said, and less than cysteamine because it gets into the tissues better. But we don't know. We need to prove that. And we're going to then, when we know the dose, we'll run it for 28 days and do various tests to make sure that it's safe in the short term. And we can then measure the volatile gases, look at the tissue levels using skin biopsies. And at the end of that, we should have enough data to persuade the regulators that we can go on and do a more extensive study. And I'm not going to, you can talk to me afterwards about what we're going to do later, but I'm not allowed to say it over the microphone. So. And this just goes into more detail of what we're going to do in the second study, the 28 days. And hopefully we'll have enough data to prove to everybody. So lastly, as I said before, CF-10 was never designed to be better than cysteamine in terms of its efficaciousness or efficacy. What it's designed to do is to make life better for patients. And therefore, we're very keen. You will hear a fantastic talk very shortly, much better than my talk, from Dr. Dwight from Hamburg about patient-reported outcomes. But we are working to design a very short questionnaire to get patients to give us their views on how much better or not they feel when they're taking CF-10. And it has the potential to be very good in terms of getting CF-10 on the market. So I conclude by saying that CF-10 potentially can benefit. It's palatable. It will be easy to take. Eventually it will be available as a tablet or capsule. And we know that there's a great need in children. And so there will be we're working on a liquid formula. So it'll go, you call it a G2, but we call it something else, but it doesn't matter. And I am allowed to say this. The initial studies will not be in babies or children because of the way the regulators work. So we will start probably with adults and then very rapidly go down to the age of 12. Rules in the UK are different and Europe are different to America. But anyway, we've got a drug. It won't cause you the gastritis. You won't have to take it very often. It'll improve your quality of life and psychological. When I looked after patients with cystinosis, I spent 10 minutes asking about their physical state and about 20, 30 minutes talking about other things. If it's better for the patients, you'll take it regularly, and hopefully you won't get all the problems associated with not taking it. I finish by just saying that this would never have happened if Ros Anderson, who unfortunately died very young a couple of years ago from a rare disease, if she hadn't bumped into John Terry, who some of you may remember, who was a cystinosis patient, he basically founded Cystinosis UK, and she got to work with her team on the CF-10 journey. And this is just the list. Nothing happens in isolation. Nothing happens without the help of a lot of people. And this is another one. So thank you very much for your attention. And I'm around if you want to chat.